So I'm the last one here. You probably heard everything so far about biomarkers and how to use them and how to commercialize and what have you. I'm going to take this just from a slightly different angle. And in my presentation, I will discuss a little bit what are biomarkers, because we have heard a lot today about maybe antibodies, proteins, or DNA or mRNA, but there are of course a lot of other biomarkers that could be useful. And then the tools available for analyzing biomarkers are also quite a large number of different techniques. Not all of them are suitable for moving into a routine clinical setting. And I will pick 6 examples of technology areas or methodology areas that I think will be very, very important for the future. And I will end with some conclusions. So if you go to Wikipedia and look what a biomarker is, it really refers to something that you measure and which is characteristic for something. Specifically then, in our case, the biological state, whether it's the normal biological process or a pathogenic process or something happening during a therapeutic intervention. So with that definition, you could, of course, see that there are a lot of different ones that could be used. So they could be used also for a lot of different things, just as pure information. You have heard about the prognostic and predictive ones. Of course, the companion diagnostic is a very new and important area related to personalized medicine, that you could actually select the patient that is given the treatment. But the biomarker could also be the drug target. We've heard examples where actually the target and the biomarker are the same, but of course it could be other biomarkers as well. And drug delivery is also an area, because biomarkers could be used as targeting the area where the drug is positioned. And there's a lot of work ongoing currently in the pharmaceutical industry where you do combination of the drug and something that finds the area where the drug should interact. Then, of course, biomarkers could be used for a lot of different things as well. Very many different stages. Just to set the biological state of something, you could look at predispositions. For instance, today we have this test which is delivered by Myriad Genetics. It's a BRCA1 and 2 test where you look for mutations. It costs $3,000. They are the only one that have this test because they have it patented and they've been fighting around it. But this one is a predisposition for breast cancer. And then you have to go through genetic counseling in order to determine what you're going to do with that type of information. We have screening tests, like Carl Bollerbeck was talking about, this screening test for high-risk groups in pancreatic cancer. But there is a lot of other things in relation to screening. The correct diagnosis, you have a symptom, you bring in the patient and you do the test. And also looking at biomarkers as a monitor of a drug treatment. You treat the patient and look what is happening. All of these things require perhaps different methodologies, different technologies, different thinking in order to really make it successful and make it into the clinic. And biomarkers, I mean, there are so many different ones. All of them are being used in different stages today. Of course, mainly DNA and mRNA is used, but we see more and more about microRNAs. We have also a few interesting cancer diseases which are based on gene fusions, and all the epigenetic factors that is involved in this process. On the protein side, of course, we have proteins themselves, but There is, of course, modification of proteins. You have fragments of proteins. You have antibodies. You have fusion of proteins. And then you have a lot of complexes, which are quite important. And I just recently went into the area of HLA, and it will take me 10 years to really understand this whole concept of HLA. And still, it's involved in almost everything that is happening. And this is an unutilized area in my world. And then, of course, all other different types of things you could look at. I mean, even temperature, the raise of a temperature is actually a biomarker in itself. And then where you find this biomarker, of course, there's so many different types of samples where you could find it. And then if you sort of combine them and looking at many markers in one area, like the transcriptome, you look at all mRNAs in that area. And of course, pathways. I mean, pathways will be extremely important in the future to understand what is happening in your biological system. And then, where do you find them? Is it the whole population? Do you have a patient cohort? Are you looking in biobanks? Are you using animals instead? Or are you using cell cultures? So there's so many different variants here to combine. And you have to select which one is the best. And technologies. You pick, these are sort of the ones that are commonly used, and there are thousands more. Which one to choose, how to choose this one. So one thing is that there are a number of questions you have to answer when you're looking at the tools. So will this tool answer the question or the hypothesis you have? Could I look at more than just one? Is there a combination of things that might give me even more information? Am I looking at qualitative results or quantitative answers? Of course, it's always nicer to have the qualitative answers. And from the world where I've been, in the immunohistochemistry world, where you have a pathologist sitting in a microscope looking with his eyes, trying to identify, yes, this is that, this is this. And it's only based on his memory in his head what this really is. There is no measure. We're moving into that space now with more image analysis, but still it's a very good example of the difference between qualitative and quantitative answer. And we also heard about the fact that could this tool that I use in my research actually end up in a clinical laboratory in the end? Some of these techniques we refer to will never hit the routine lab, either because it's too expensive or it's not really feasible to have there. So is the tool also commonly used and accepted? There are actually a number of biomarkers that have not been accepted based on the fact that you've used a technology to generate that information that people don't really like. And you're probably not expert in everything. So even on the clinical side that you need to have your collaborators, you also need to have collaborator on the tool side. Find the right people that know something about it, and you will avoid a lot of trouble later on. Sampling, one thing is of course that the less invasive you have your sampling method, the better it will be accepted. But it's really, you have to really find out how to do this. And then, could I afford this? Some of the things are, of course, very, very expensive, and therefore one has to really think about it. So, I'll jump into a few examples here of tools and methodologies that I think will be extremely important in our space. And the first one is next generation sequencing. And this is actually one of the latest tools now that is going to come out on November 25th. This is a DNA sequencer. It's called Minion, and you connect it to your computer like a USB stick. The first version of this one will be for DNA sequencing, but it will also be adopted for RNA and for protein analysis. And it's based on nanopores in a very intelligent way. This is now going to be released to the market as an early access program. And if you as a researcher go to Oxford Nanopore and you'll deposit $1,000, you will get a machine, and for that you will report your data around it, and you get a subsidized reagent piece to this. And when you return it, you get your $1,000 back. It's stated that this sequencer will cost $900. And this will probably change the world of how you look on DNA sequencing. You could do it at home. It's just amazing how technology develops in this space. Another technology area, which is of course related to sequencing as such, but it's also related to the fact that you need to deal with the data. And the amount of data that is generated is just increasing enormously. And I just represent this in a way with how much it costs to sequence a base and how that has developed. And somewhere here, a number of years ago, we broke the Moore's law in that everything doubles up every 10 years, and now we just left that. And today we're talking about, I mean, sequence for less than $1,000 for a whole genome. It's quite amazing. The other technology area, which will, apart from being sort of a research tool, now will move into the clinic, is mass spectrometry. And there is a lot of new advancements on the mass spectrometry side, represented here by Thermo Fisher Orbitrap technology. These types of mass spectrometers now are capable of having a resolution of 0.001 Dalton. It means that you could distinguish any molecule, even on the protein side. And this type of machine could actually more or less do anything. It could do proteomics, you could look at glycosylation, you could look at anything. And these are now coming out in clinical versions, so they're moving into the clinical lab. So more and more assays will actually be used on mass spectrometers in the future. In vivo imaging, to not do any invasive thing at all, but actually look in the body. And in vivo imaging will also be connected with biomarkers in the sense that you might inject a biomarker and then do the in vivo imaging and see exactly where you have your problem. And what is already done now is that you actually combine your biomarker then with a drug instead. So when you localized it where it is, if you do surgery or if you add your drug of interest to hit that area, and then you look how it's developed. So imaging will be sort of a part in a lot of a tool that will be used in this world more and more. And there's a lot of development happening, and we'll see more things coming out in this field. I call it cell-based assays or functional assays. There's actually a requirement or a strong advice from FDA that anytime you come up with a new drug, that you also have some functional assays connected to it. And cells are, of course, a very interesting tool. And there is a lot of things happening now on improving the cell-based technologies, specifically in one area to do 3D cell assays and not have the two-dimensional cell assays, and also how you modify these cells. And stem cells are, of course, one important tool in this area where you could do a lot of interesting things. But one of the perhaps most interesting areas are immune cells. And for instance, what is now being done on dendritic cells, but in order to use the immune system to actually help you in a lot of areas. And immune system will probably be one of the most important things in relation both to biomarkers but also for drugs in the future. And immune system is very interesting in the sense that it actually is a precursor to anything else, because it's the immune system that first starts to react when you have a perturbation in your body. In the immune system, you will find a tumor or a cancer much earlier than it manifests itself in terms of you having symptoms or anything. And one of the examples was the thing that Carl Borbeck referred to in his test, because the biomarkers that are actually chosen for the pancreatic cancer test is immune system biomarkers. So that's how the selection was done in terms of finding this. And also in terms of using all these systems together with gene therapy to actually introduce any other types of gene into the system. So cell-based assay and cell therapy is a very important area for the future. Drug delivery, I just picked out of Wikipedia. And why am I saying drug delivery? Well, drug delivery today is about how you deliver your drug into the body. And some drugs that are developed are so toxic that you can't use them. So you have to find alternative way how to make them less toxic, or reduce the dose but target them to the area where it's going to have its action. And today, drug delivery also is a lot about nanochemistry and nanotechnologies, because nanoparticles have been started to get used as a tool to find a tumor, because nanoparticles at 10 nanometer level are penetrating into cancer cells through the membranes, because the membranes are different than in the normal tissue. So you will actually accumulate nanoparticles in a tumor. And combining that with a biomarker that finds the area and combine that with a drug. So with the three components, a nanoparticle, a biomarker, and a drug, we have a completely new way of how to treat cancer tumors. So drug delivery as a component in thinking of what you're doing, that eventually it's going to be a drug, and how could I use my biomarkers in this context. Big data. Every day around 2.5 exabyte data is generated. That is 2.5 times 10 to the 18, 1 billion terabytes every day. With everything that we're generating in terms of information today, the common way of how to deal with data is not really possible anymore. You have to do completely different thinking in how you do your algorithm, and you have to have massively parallel software running on tens, hundreds, or thousands of computers at the same time. Just recently, US government released $200 million to evaluating how to handle this from a US national perspective, because this is getting so huge. And you will see companies like Google and Facebook and all of them being involved in this process in the future. And sometimes you feel a little bit humble when you look at this and try to understand what the complexity will be in this and how to make use of this type of information. But big data is definitely one extremely important area to be involved in. And for us working with biomarkers or information about the patient, I mean, this will be an important asset for the future. But also for clinicians in terms of being able to extract data, what has happened with similar type of patients in other parts of the world, and how to be able to retrieve that data. Just a few words also on that. Some time ago, you could say you were developing a biomarker assay. You had an antibody in a tube and you poured it into some kind of plastic well. We moved into automate this process because we had so many samples. So you had all these big, huge clinical automated systems. Today, we're talking more about solution-driven situation in that you deliver a complete solution to the user, and therefore you have to think about a lot of different things in order to make that solution complete. But I think the future will be even more information-driven. So you have both the solution in terms of hardware and reagent and what have you, but the information part, how to connect the thing that you're delivering, will really be important. And for us in the biomedical field, the electronic medical record, the information about a patient will probably be the most important information source that we can have. And specifically if you work in an area like diabetes, I mean, that kind of information is essential in order to make the right conclusions. Then one has to also think what is happening in the lab. And today, you could say that all labs have less and less hands to do their work. Everyone is cutting down budgets, and all the labor-intensive work processes, in one way, people want to get rid of as such. And all of this put a high pressure specifically on the diagnostic industry in terms of what they deliver, because it's not always easy to get something into the lab and get it reimbursed. Today, US have made a decision to turn around their whole reimbursement system. So the CMS, who is the authority that is coding every new product that is coming out on the market, have taken away all the codes. And they were indecisive for 6 months in 2013. So from January to June 2013, every lab that had a molecular diagnostic test did not get any reimbursement. And it turned out that a number of labs actually went bankrupt and is no more. Now they have given them a new code, but there are currently 450 companies that have their product under review in the CMS system to get reimbursement. And they're picking the ones which are most interesting. So a lot of companies are today waiting to be able to get their assay out on the market. And they have changed two things. They are now requiring you to show clinical utility. And they also require you to show health economy. So with a test like Kirsten presented here, where we're still waiting for the clinical utility, they would have not today approved that test for reimbursement. So there is a lot of things you have to think about. And of course, you could never, ever skip quality. I mean quality in your product in the end. Because if you fail with your product and you get bad reputation, or you get a warning letter from FDA or whoever, you really need to see to that you follow your quality measurements. So when you're selecting how to do with your biomarkers, there are a lot of things you have to think about. And there's not always that much help to gain, but there's a lot of gain to read what others have done, and specifically don't repeat the mistakes that others have already done before. Then, as an end up, I just use a citation from Dr. Zolg, who was the head of proteomics at Roche. And he said that, well, in general, looking at it, you would need something like 1,500 samples to test your candidate biomarker in. And if you don't have that, you don't really have a convincing fact that the diagnostic industry, clinical chemist, and physician, that they believe that you have something useful. And the approximate cost for coming to this final validation is around $4 million today, if you include the clinical utility and the health economy study. And this is one of the reasons why we have difficulties with venture capital companies, because one ticket to have your product out is starting at $4 million. For instance, one of the newly formed companies, Foundation Medicine, just made an IPO. They're only 35 people, and they made an IPO on $106 million. They have a market cap of $900 million for this company, and they're doing DNA sequencing. They now release a product which is a sequencing test with 181 cancer genes, and they're selling this test for $5,500. But the payer organization requested them to show clinical utility in a prospective study. So they have to run a couple of thousands of samples prospectively now for the next coming 3 years. Under that period, they get only reimbursed with half of the price. And they have calculated now that they have to spend $12 million on these studies in order to get their test approved. So we're talking about completely different money today than we did before. And the whole thing becomes more and more complex. So you need to have partners. You have to have smart people around you. You have to connect to the smart guys so you're doing the right thing. Because no one is going to be able to do something on their own today anymore. So, that was that. Thank you.